one of these hot and upcoming topics. So take it away.
Thanks. Hi, I am Abhishek Roy from University of Massachusetts Amherst, and I will present our work on massively parallel processing of whole genome sequence data. In the last 15 years, the sequencing technology has advanced significantly. The amount of sequence data has doubled about every 7 months, far exceeding the Moore's law for computational power. The first whole genome was sequenced in 13 years at a cost of $3 billion. Now, we can sequence the whole genome in 2 to 3 days at a cost of few $1,000s. The sequencing centers are generating terabytes of data each day. Each sample contains half to 1 terabyte of data. This has transformed genomics into a new paradigm of data-intensive computing. The advances in sequencing technology and the large volumes of data produced allow us to answer biomedical questions with unprecedented speed and resolution. It also helps in realizing the vision of personalized genomics. Personalized genomics would allow drugs to target precisely the different stratifications of patients and diseases. First, I will give a brief overview about the sequencing process. Human genome consists of 3 billion base pairs. Each base is a character A, T, C or G. First, in the lab process, a test genome is broken into multiple small fragments. Then, a paired end sequencing machine reads the fragments from both the ends. These reads coming from the same fragment are called paired reads. The gap between paired reads is known for different sequencing machine. Each read is a sequence of 30 to 1000 bases. At the end of this process, billions of short reads are generated per sample. Each base in the test genome is read on an average 30 to 120 times. The first computationally intensive part of the pipeline is the alignment process. The alignment process maps the paired reads onto a reference genome. One read can map to one or multiple positions on the reference genome. Then the reads undergo a series of steps to remove errors and finally detect the variants. In this example, we show a set of reads of test genome over a segment in the reference genome. The majority of the reads support the variant called G to T. For human genomes, there are millions of such variant calls. Throughout this pipeline of steps, the data is transformed into different formats by the different processing steps. First, the sequencing machine generates the raw read sequences. In these reads, the reads belonging to the same pair share the same read name. Also, there is a quality score associated for each base of the sequence, which is the likelihood of that base call being incorrect. After alignment, multiple attributes are added to each record. These attributes include the coordinate position on the reference genome, mapping quality, etc. These set of reads are finally processed to detect variants. The variants record the position on the reference genome and the difference with the reference genome. Overall, the data processing pipeline can be partitioned into 3 phases. The first phase, primary analysis, generates short read sequences. Then the billions of short read sequences go through secondary analysis. It includes over a dozen steps for alignment, data cleaning and variant detection. It also reduces the volume of data from billions of reads per sample to millions of variants per sample. This phase can be called the ETL process of the genomic data processing pipeline. The secondary analysis phase is very resource intensive and it can cause significant delay in processing times. In our work, we aim to reduce the turnaround time for secondary analysis. The detected variants are then loaded into databases and evaluated, and further population scale studies are performed as part of the tertiary analysis phase. The secondary analysis phase has major computing challenges. First, the data needs to go through a complex analytics pipeline with 15 to 20 steps. Users can customize pipeline depending on sequencing technologies, species, and diseases. This pipeline is very long running. Current pipeline can take 3 to 23 days to finish. Most of the algorithms were in genomic data processing were not designed to efficiently process large sets of data. Majority of algorithms offer either no parallelism like mark duplicates or limited parallelism like the multi-threaded alignment BWA. In scatter-gather parallelism, the user manually partitions the data into smaller partitions and collects the results at the end of execution. However, the scatter-gather parallelism is not sufficient because it allows only a limited degree of parallelism. For example, if you partition the data by chromosomes, we can get only 23 partitions. Also, increasing the degree of parallelism would increase the error rates. It is the responsibility of the user with scatter-gather parallelism to keep a track of the errors. Our pipeline aims to automate both high degree parallelism and managing the error rates in the pipeline. To solve these challenges, we decided on a set of objectives with New York Genome Center. We want to design a big data platform to reduce the processing times from weeks to days. We also want to have a thorough understanding of the big data technology and its strengths and limitations when applied to genomic data processing. We would also like to identify key research questions to improve the big data technology itself. Our pipeline should meet the following requirements. It should support customizable and varied analysis pipelines. Genome centers cannot re-implement the many analysis steps to port them to different parallel platforms and subsequently perform an accuracy check. Our platform would support parallelization of analysis programs in their original implementations without having to rewrite them. We also cannot assume that the data fits into the memory due to sharing of resources among concurrent pipelines and analyzing batches of samples. Now I will present the design of our big data solution called Giselle. The key feature of our architecture is wrapper technology. Wrapper technology allows us to reuse existing genome analysis programs in a big data framework. It has several layers of software which can be used to wrap existing data analysis programs. First, the data in the standard file formats are uploaded to a distributed database. This splits the data into small blocks and breaks the file format requirements. When an external program like BWA tries to read these blocks, it would fail because data that meets the logical conditions is not at all accessible. To solve this problem, we determined logical partitions for each analysis program. Our storage layer supports logical partitions and includes optimizations like co-location of data. These blocks are also not in the required genomic standard file formats. To transform these blocks to correct file formats, we have built a runtime layer which includes the data transformation module. The data transformation module at runtime converts these blocks into the correct file formats. Then for different analysis programs, we determine the correct partitioning schemes. We analyzed the data access patterns of the many steps in the pipeline and found several common data partitioning schemes. These partitioning schemes are implemented in our Genome Data Parallel Toolkit. An external program can use a partitioning scheme from this library to access the data in a logically correct manner. Later on in the slides, we will show that in spite of using correct partitioning schemes, we can still have errors in the results. For the keeping a track of errors in the pipeline, we have built a error diagnosis toolkit. All of these layers are detailed in the paper, but for this talk we will focus mainly on genome data parallel toolkit and the error diagnosis toolkit. For genome data parallel, in genome data parallel toolkit, we offer a collection of different data partitioning methods. The simplest access pattern is grouping data by a logical conditions. These steps, these are some of the steps which group data by a logical conditions. One of these steps, BW alignment, aligns the reads of a pair together. Recall that reads of a pair share the same read name. So, to achieve this data access pattern, we can just group the data by read name. Range partitioning employs a data access pattern in which reads are considered as intervals over the reference genome. Range partitioning can be static. In static range partitioning, the reference genome is divided into predefined segments. Each partition consists of all reads overlapping a segment. Some examples are when you partition the data based on chromosome or known set of genes. Range partitioning can be dynamic as well. In this, the external analysis programs determines the partitions as it scans the read data set. Our pipeline supports both static and dynamic range partitioning. In compound partitioning, two grouping conditions are applied on the same data set. One of the examples for this is mark duplicates, which we will discuss in the next slide. Mark duplicate flags the duplicate reads. Duplicate reads are an artifact of the DNA preparation method, and they might introduce biases in the final variant call results. Duplicate reads have the same position, sequence and orientation. This figure shows 3 read pairs. In this, for each read of the pair is successfully mapped to a position on the reference genome. These read pairs are called complete matching pairs. For R7, the other read of the pair is unmapped. The R7 is part of a partial matching pair. The mark duplicates algorithm applies 2 different duplicate detection criteria on complete matches and partial matches. For complete matches, the reads belonging to complete matching pairs are compared with other complete matches only. The gray boxes shows the partitions for detecting duplicates in complete matches pair. These partitions were made from a compound key derived from the positions in both reads of the pair. In this example, between the pairs R1, R2 and R3, R4, the one with lower quality will be marked as duplicate. For partial matching pairs, the partial matching reads are compared to individual reads in both partial and complete matches. In this example, the partial matching read R7 will be marked as duplicate because it coincides with reads R2 and R4 from complete matches. Our partitioning scheme generates these partitions automatically from the read data set. These partitions can be processed independently of each other, thus allowing us to leverage data parallelism. These partitions can be passed to a mark duplicates program which would detect duplicates in these partitions. Because we applied 2 grouping conditions on the records, one read can be replicated in multiple partitions. In a distributed environment, this increases the amount of data shuffled. As in optimization, we keep a track of partitions where partially matching reads are present. This allows us to apply the second grouping condition only on those partitions where the partial matches are present. This reduces the number of partitions and the amount of data shuffled. We arranged a pipeline of different analysis program into a sequence of map reduce rounds. The external analysis programs are denoted in the green boxes. We also use different partitioning schemes from our Genome Data Parallel Toolkit. These partitioning schemes are implemented by using MapReduce Shuffle. This pipeline will serve as the testbed for our evaluation. The aim of our evaluation is to answer three questions. Does big data technology offer superlinear speedup? Does big data technology offer sublinear speedup, and why? And what is the quality of results variants, given that these variants are human mutations which will be used for diagnosis? To compare the performance, we evaluate our pipeline against the GATK best practices, a multi-threaded pipeline known as the gold standard for genomic data analysis. GATK best practices runs processes on a single node with multi-threaded versions if available. We use the following metrics to capture the performance. The first is the speedup over the single node program. Please note that the single node program is the equivalent version in the GATK best practices and can be a multi-threaded program as well. The second metric, resource efficiency, measures how effectively the additional resources are used. We will define sublinear and superlinear speedup in terms of resource efficiency in this talk. Superlinear speedup corresponds to resource efficiency greater than 1, and sublinear speedup corresponds to resource efficiency less than 1. This is the summary of our performance number. We ran the pipeline in a cluster with 15 data nodes, and we can run up to 90 parallel tasks on each in the cluster. The first and third column are the runtimes from the single node execution and the parallel execution. As you can see, observe that we are able to significantly reduce the running time. For example, for mark duplicates, we were able to reduce runtime from around 26 hours to approximately 1 hour. We are also able to achieve superlinear speedup for alignment. The other steps show sublinear speedup, and in the next few slides we will investigate the reasons behind them. The BW alignment program is a CPU intensive multi-threaded process. This figure shows the sublinear speedup of BWA as we increase the number of threads. The BWA program has many bottlenecks, including a synchronization point for reading the data from files. However, with Yarn's process thread hierarchy, we are able to run many processes with few threads rather than one process with many threads. This allows us to have a superlinear speedup compared with the 24-threaded BWA as the baseline. Now we will investigate the reasons behind the sublinear speedup. Let us consider the program mark duplicates, which uses the fine-grained compound group partitioning schemes. We are able to run mark duplicates in up to 90 parallel tasks in our cluster. Another program, haplotype caller, which detects the variants, has a coarse-grained partitioning scheme based on number of chromosomes. So we can run only 23 parallel tasks. First, we have to pay the overhead to repartition the data. Then the haplotype caller with 23 parallel tasks took 7 hours to finish. Even though we had the resources to run up to 90 parallel tasks, our resources were underutilized because of the coarse-grained partitioning scheme of haplotype caller. This change in degree of parallelism impedes the utilization of the resources in our cluster. Another reason is the data shuffling cost. Different partitioning schemes are implemented using the MapReduce shuffling. For example, for mark duplicates, partitioning took 35% of the runtime. We also have to convert the objects between the Hadoop formats and the correct genomic file formats. This copy and convert overhead by the runtime layer, data transformation layer, is itself 12% of a running time in a map task. We also observed that in our parallel implementation, the external programs are called on small batches of data. We observed that for some programs, the sum of running times in the parallel implementation is more than the running time in the single node execution. This is because multiple reasons, including fixed startup overheads of the program, cache behavior, etc. Now we will move from the performance evaluation to the accuracy validation. We observed that many parallel programs in the pipeline, like BW alignment, produces different results with serial and parallel execution. These differences of individual steps are propagated in the pipeline and affect the final variant calls. This raises two questions. What are the differences between results of the serial pipeline and our parallel pipeline? And are the results of our parallel pipeline acceptable? From our discussions with the bioinformaticians at the New York Genome Center, we found that the ultimate accuracy measure is the impact on final variant call. Using our error diagnosis toolkit, we can first construct a hybrid, we can construct a pipeline up to any step we want to analyze. For example, in the hybrid pipeline, if we want to analyze the impact of mark duplicates, we construct a parallel pipeline up to mark duplicates and then run the remaining steps in a serial manner. Then we compare the final variant call outputs between the hybrid pipeline and the serial pipeline. This shows the comparison between variant calls from serial and hybrid pipelines. We observed that 99.8% of the variants are common to both pipelines. The analysis of variant calls using a set of biological metrics also revealed that the variant sets differ only in low confidence calls. One of these metrics is the read depth metric. We observe that the read depth for variants which are common to both the pipelines is around 71, which is close to our sample read depth of 64x. The hybrid and serial pipelines have very high read depths of close to 270. These read depths correspond to artifacts in the alignments and come from regions in genome which are hard to map and are very repetitive. Thus, we can conclude that our parallel pipeline does not increase error rates or reduce correct calls. We also analyzed the differences in alignment from the output of the single node program and the parallel pipeline. We found that the majority of disagreeing reads are gathered around hard to map regions. This figure shows hard to map regions in these red and black stripes. The disagreeing reads, majority of disagreeing reads falling in these hard to map regions are shown by the spikes in the blue lines. These hard to map genomic regions have low mappability and are very highly repetitive. These disagreeing reads have low quality score as well. Thus, the difference in variant calls can be attributed to such hard to map regions where both serial and parallel versions give low quality results. To summarize our work, we will move to our study of the both performance and the accuracy resulted in many different areas for future research work. One such area is automatically safe partitioning existing programs. Currently, finding the correct logical partitions for different analysis program is a very manual process. How do we automate this is a question for future work. We are also extending Genome Data Parallel Toolkit to include more complex partitioning schemes. In error diagnosis, we can also use provenance to track errors in the pipeline. However, tuple level provenance requires substantial code change in each object creation line. How can we use things like mini batch provenance depending on the access pattern is a question for future use. I will stop this presentation with these future work and take the questions now. Thank you. definitely offers higher level of abstraction primitives. But one of the issue with this pipeline is we want to reuse the external analysis program. We don't want to rewrite them using Spark RDDs. And also in-memory assumption can fail in some cases, especially when you analyze batches of samples like parent and child and different samples together. In that case, the data volume can be in few terabytes.
Sure.
So that is one issue, but yeah, it can be used as an underlying framework, but overall the same questions will be present.
And maybe as a follow-up to that, so imagine like someone comes up with some funky new algorithm. I know like the variant graphs are really hot right now. And like if they say, okay, from the ground up, we're going to implement things on top of your framework. Do you think like, would it make any difference for them in terms of development?
Yes, that would make a lot of difference because existing genome analysis programs are not very efficient.
Right.
If they design the programs keeping in mind the correct data partitioning requirements, anticipating the needs, or even providing us hints so that we can implement partitioning schemes for them, then it would be very useful.
And those hints would be partitioning hints.
Yeah, partitioning hints. And in generally, because you observe that we have very low resource efficiency, a major part is because these algorithms are not very efficient.
Last chance. All right, cool. Thanks a lot.
Thank you.